Let's talk about worms. Not the earthworms living in your garden, but helminths, worms that are parasitic to humans. Helminths can cause a wide array of disability, and they infect almost 2 billion people worldwide. Yes, billion, with a B. If you've never heard of these pervasive infections, it's probably because they primarily occur in impoverished areas of the tropics. For infected individuals, we only have a few treatment options, and worms are becoming resistant to them. We desperately need new treatments. My thesis aims to find new sources of antiparasitic drugs, but where do we find drugs? Was your first thought CVS or Walgreens? Me too, but historically this wasn't an option for infected individuals, and instead they looked for treatments in native plants and animals. This might seem odd to us pharmacy goers, but nature actually has an advantage when it comes to creating drugs: evolution. Living things have evolved to produce chemicals called natural products that can ward off enemies and protect against environmental stressors. Scientists have used these highly specialized natural products to create many of the medicines we have today, including penicillin, aspirin, and even the antiparasitic ivermectin. I'm looking for new antiparasitic natural products in microbes like bacteria and fungi, because microbes and humans share a common enemy: worms. Microbes and worms compete for the same environments, and some worms even consume microbes as food. If your neighbor was trying to eat you, I'm guessing you'd find a way to fight back. So we predicted these microbes are producing antiparasitic products. Now that I know where to look, I can perform 3 steps to begin the drug discovery process. First, I collect. I gather microbes from places where worms also live. I grow the microbes and I collect all of the natural products they're producing in a sort of chemical soup. Second, I screen. I expose the worms to the chemical soups and I take hundreds of photos of them, deciding if they're paralyzed or developmentally delayed, anything that could imply antiparasitic potential. Third, I analyze the promising chemical soups. I profile them, looking at every natural product present and deciding which one harmed the worms and if it has promise as a treatment. So far, I've set up hundreds of these worm versus microbial soup matchups, and in dozens of them, the microbial products won. Even though we've identified some antiparasitic natural products, most, if not all of them, will fail the rigorous process required to become safe drugs for humans. But for now, this research has shown the importance of thinking creatively when addressing a problem as huge as parasitic infections. It's only with the combined knowledge of medical history, biology, and chemistry that we could develop this strategy for finding new antiparasitics. Thank you.